All right, class, let's get started with our discussion on how to diagnose infections. This chapter is going to focus primarily on the three big methods of diagnosing infections. We'll have genotypic, we'll have phenotypic, and then we'll have immunologic methods. And right now in the field of microbiology, we're seeing a big shift, a big change right now. 50 years ago, everything was all about the phenotypic methods. We're all about the classical wet chemistry techniques, the staining techniques, so to speak. And then the biotechnology revolution happened, and we started using the genotypic methods to classify our microorganisms. And now we're also adding on to that our immunogenic methods of classifying our microorganisms. So without further ado, let's get started. We need to talk in this microbiology class about many different diseases that afflict human beings. That's one of the main emphases of this class, the entire second half of the class. Now, to identify what the specimens are, what disease we're working with when a patient comes into the clinic or the emergency room, you'll be using those three primary methods, our phenotypic, immunogenic, and genotypic methods. Our phenotype consists of the traits that are being expressed. So what's the shape, what's the size, what's the morphology of our microorganisms. Our immunogenic is primarily going to depend on a serological analysis that is dependent on antibodies. And then finally, our genotypic method is going to rely heavily on DNA analysis. And the workhorse of our genotypic methods are the thermocyclers performing polymerase chain reaction and then dumping that bad boy, that DNA, into an electrophoresis chamber to separate out the DNA. Based on data from all three of these fields, we can then identify our unique bacterium or other microorganism. So here's a nice summary figure. When we look at phenotypic methods, we look at the color that this bacterium is stained, its shape. When we look at our genotypic method, we're looking at how the DNA fragments are arranged and what DNA fragments we have. And then finally, with our immunogenic methods, we look at whether or not the antibodies will stick. If we look at our genetic means of identification, this right now is the gold standard. This can stand alone, and most labs right now rely on rapid PCR and genetic analysis. Traditionally, with our old-fashioned techniques, we would use those biochemical assays, so whether or not the bacterium would live in the presence of catalase, or whether or not it had produced coagulase, or whether or not it was gram-positive or gram-negative. And there are some newer methods that are now being utilized, such as CT scans, magnet MRIs, and PET scans as well. These three techniques usually are going to be used in conjunction with the other two methods. Obviously, light microscopy is a bread and butter technique for identification within the laboratory. The light microscopes represent a relatively low investment or low cost investment and are still going to be excellent for giving us some just basic morphologies like our cell size, shape, and arrangement. Light microscope can tell you the difference between a staphylococcus and a streptococcus, and that test costs almost nothing. We obviously can still do lots of gram stains and doing our other endosporin stains, capsule stains, acid-fast stains. Most of the staining techniques that you've either done in the digital labs or that you do with the face-to-face labs are still used in a lab setting. We've supplemented the light microscope, though, with the electron microscope. Our electron microscope, instead of relying on beams of visible light, relies on beams of electrons. And since beams of electrons have much lower wavelengths, or much shorter wavelengths to them, those shorter wavelengths mean that we can see more detail and can help us identify cell wall, flagella, structural features of the bacterium. We still could do staining techniques, but an electron microscope is more reliable and quicker than the staining techniques to identify these cellular appendages. If we look at our macroscopic morphology, these are things that we can see with the naked eye. Macro, meaning big. So the big shape or big picture, what can we see with the naked eye? So what is the color of the infection? Is it a black infection versus a greenish infection? That's a very important bit of information. Black, green, yellow, or white or bright red. All of those colors for the oozing growth or sore on the patient can be very important. Or how quickly is it growing? Or how does it grow in broth versus gelatin versus agar? Other traditional phenotypic methods include enzyme production or biochemical assays. So this is a good example of this would be taking your bacterial sample and exposing it to a couple drops of hydrogen peroxide. Does it produce the catalase? So these enzyme productions that we can use simple biochemical assays for are still used fairly regularly because they're cheap, they're quick, they're easy, and they're reliable. We also have diagnostic tests for determining the presence of very specific enzymes. So is that bacterium going to ferment lactose, or is it going to ferment sucrose, or glucose, or galactose? What sugars can it live off of? What complex polymers can it digest? Can it digest plastic? Can it digest cellulose? Can it digest glycogen? What gases are produced? And how sensitive is that colony to an antibiotic, and which antibiotics is that colony sensitive to? Is it sensitive to a broad-spectrum antibiotic, an antibiotic that focuses on gram-positives, or an antibiotic that focuses on gram-negatives? These are important diagnostic criteria. So for our chemical analysis of our phenotypic methods, we're going to look at the specific structural substances that are present in our microbes. So what kind of peptides, what kinds of short amino acid chains are in the cell wall, and what kind of lipids are present within the cell membrane? Are there lots of phospholipids relative to the amount of cholesterol, relative to the liposaccharide or lipopolysaccharides? So the lipid profile or the peptide profile also can be very enlightening. Many times what you'll do to isolate this information is culture the bacterium, grind it up, smash it up, and then run its bits and pieces through a gas chromatograph, a mass spectrometer, or a, I'm drawing such a blank here. A gel column. There we go. My bad. Sorry about that, class. Now, if we look at our genotypic methods, these are the current gold standard right now. Our genotypic methods are wonderful in that one test does it all. So instead of having to perform many individual little tests, we can have one test that gives us all the answers we need. Now, there's pros and cons to the genotypic method versus those other phenotypic or chemical-based methods that I previously talked about. The genotypic method means that we don't always have to necessarily culture the microorganism. We can work with that small sample of DNA that we have in the small samples of microorganisms we already had. Our genotypic methods are becoming increasingly automated in producing faster and faster results that are almost always more precise than our phenotypic methods. But if you're looking for one specific characteristic, maybe the presence of a catalase enzyme, or whether it's gram-positive versus gram-negative, if there's just one little tiny bit of information you're curious about, then our phenotypic methods or our biochemical methods still are going to be preferable because that one individual test does not take that long. Genotypic methods typically are going to take a couple hours. So we're looking at multiple hours for the genotyping, whereas each individual phenotypic or biochemical assay is on the order of minutes. We can look at a viable non-cultural state of a microbe. The viable non-cultured microbe is a microbe that's still alive but hasn't been actively reproducing in a laboratory setting yet. These microbes cannot be identified by genotypic methods. One of the reasons for this is because we eventually need to have a nice pure culture of our microbe to use as a control or to compare our genotypic information to. And if we can't get the microbe to be cultured in the lab setting because it's viable but non-cultured, we can't use our genotypic methods for it. Our immunologic methods rely on antibodies. So how those antibodies, think of the immune system and the production of antibodies by the plasma cells, which are progeny of the B lymphocytes. Think of that antibody response. Depending on whether or not the antibody sticks, we can say whether or not something is present. So what we need here is a specially designed antibody that's going to stick to our favorite molecule, whatever our favorite molecule is. And if the antibody can stick to it, the sample, then we know that our favorite molecule is present. Now, we also are going to need to know whether or not the antibody does stick. And it's kind of hard with the naked eye. So we're going to use some laboratory kits and some chemicals to make the antibodies glow bright green or glow red so that we can look at the color changes to know whether or not the antibodies are sticking. This is typically going to be easier than testing the microbe itself in a genotypic method. So this is going to be quicker than our genotypic methods. But on the flip side, it's also going to be more precise than our phenotypic methods. So our immunological methods are kind of a happy medium between the genotypic and the phenotypic. So, concept check class. A Gram stain of a sputum sample from a patient is an example of a blinkety-blink method of microbial identification. Would it be genotypic, immunologic, phenotypic, histologic, or none of the above are correct? So go ahead and rewind the video, find an answer, pause it, flip back in your PowerPoint slides, or check your textbook. Get me an answer. 1, 2, 3, 4, 5. The correct answer is C, phenotypic. The cell wall composition is what determines the outcome of the Gram stain. And because the Gram stain is going to look at the composition of a physical structure, we call that a phenotypic method. If you have any questions, please feel free to post them in the discussion board or shoot me an email. Happy studies!